Hello, my name is Jackie Arnold, and I have been working with graduate student Jordan Chapman and Professor Sarah Saladino for the immobilization of enzymes within a hyaluronic acid hydrogel. Now, I'm first going to describe to you all what enzymes are and exactly what it is that they do. Enzymes are proteins that help to encourage certain reactions to take place in a shorter amount of time. So these reactions are essentially what run the biological world as we know it. And there are two key pathways that enzymes can take. They can either take larger molecules and break them down into smaller components, or build up these smaller components into larger molecules. So you can see this in the case of when you digest your food. Those carbohydrates, lipids, and proteins that you eat are going to be converted by enzymes into heat, energy, and cellular building blocks. And then from here, these nutrients are going to be built back up into the macromolecules that make up who it is that you are. Now, the way that enzymes quicken up these reaction processes is by decreasing the amount of energy that you need to invest within that system in order for the reaction to occur. So as you can see here, when this substrate or this starting material needs to be created into this product, if it doesn't have an enzyme to help it, a lot more energy is going to need to be put into that system in order for that reaction to occur. Now, enzymes have oftentimes been referred to in their ability to help you digest foods, but there are a lot of other applications of them as well. Just to name a few, they're used in the medical, food and material production, detergent, and waste management industries. But again, enzymes are used in essentially every area of the society that you know and love. To describe to you what enzyme immobilization is, I am going to ask you to describe a floor, or to imagine a floor of office workers. If you take these office workers out of their natural environment and you put them, say, in the hot sun, their productivity is naturally going to decrease. So to fix this problem, you need to build them cubicles to protect them from these harsh environments while encouraging them to stay in one place while they do their job. That being said, these cubicles can't completely block off the workers from the outside environment because they'll still need to be able to receive those assignments that they need to work on. So you need to build kind of a window of sorts through which that work can travel. That being said, if that gap is too big, if you build like a doorway instead of a window, then the workers might start to begin to leave. And some will still quit along the way, but the goal is to maximize the productivity and efficiency of this floor of workers for a controllable time and purpose. And in this regard, enzymes are exactly like these office workers. In the chemical, biomedical, and environmental industries, researchers are harvesting and utilizing these abilities of these enzymes and taking them out of their natural environments and placing them in these new synthetic ones that have new temperatures and acidities that the enzymes aren't really used to, so they stop being as productive. So you have to build them kind of a protective layer around them. However, this protective layer still has to enable the enzyme to have access to that outside environment so that they can still bind with the substrate in order for a given reaction to occur. So in short, these enzymes, or our little workers, need to be controlled in order to be as productive as possible. This summer, I completed a review on a series of enzyme immobilization technologies in which these synthetic microenvironments were used to optimize the abilities of enzymes. And here are just a few examples of ways you can immobilize enzymes. The first is encapsulation. So you have kind of a capsule-like structure where on the inside are the enzymes. You can have physical adsorption, which is kind of like the sticking of these enzymes onto the surface. You can have entrapment, which is where you have kind of a net or web-like matrix that can catch the enzyme within it. You can also have chemical bonding. So here's ionic bonding, where the enzymes and the surface are oppositely charged, so they attract one another. You can also have covalent bonding, which is stronger than ionic, and that involves the sharing of electrons between the enzyme and the surface. And lastly, you can have cross-linking, where the enzymes are chemically bound to each other, and that enables a matrix that's a lot more easily manipulated by researchers than just having the individual enzymes. Now, enzyme immobilization is being used all over the world for a ton of different applications, but there are a few knowledge gaps that continuously kind of pop up. The first is being able to balance the stability with the activity of this enzyme. So going back to our office workers analogy, this is like saying we have to find that right size of a window. The enzymes have to be protected, but they also have to be able to have access to the substrates. In the case of biomedical engineering, these systems have to be biocompatible and biodegradable in order to be safe for both your body and the environment. Also, if you want to be able to ship or store these systems in any way, then they'll have to be able to continue to be reactive for a longer period of time. We also want them to be able to be reusable. So if these enzymes are just kind of one and done, they only have one reaction and then they don't work anymore, that's not very effective. And lastly, if we want to really reap these benefits of enzymes on a large societal scale, then we're going to have to be able to produce these systems on a large scale as well. So that's kind of a consideration we have to make in the lab, is whether or not these systems that we're doing, these methods that we're using, are able to be scaled up. Now, it's important to note that no immobilization technique works for every enzyme. The term enzyme includes so many different biocatalysts with all these different properties and characteristics. So because enzymes are so variable, the immobilization techniques have to be variable as well. Hyaluronic acid, which I'll refer to as HA, is currently being used as a platform for enzyme immobilization. This compound is found within the cells, or in between the cells, of nearly every living given organism. And they are being used for their biocompatible and biodegradable characteristics. HA also has these anti-inflammatory, anti-aging, and tissue regenerative properties that are very helpful in biomedical applications. And they help just generally in cellular signaling and motility, as well as that lubrication that you see in your eyes, joints, and skin. HA is highly hydrophilic, which means that it really likes water. So it can absorb and retain moisture really well, which again is a very helpful characteristic in the biomedical industry. Now, I know a lot of you have probably heard of hyaluronic acid on commercials recently because it's being used in a lot of serums and creams because of those anti-aging and kind of hydration properties that it can lend your skin. But there are a lot of other applications of it as well. And so here are a few that I read about this summer that I just wanted to share. On the left, you can see both a sprayable and injectable HA solution that is able to serve as a surgical glue and a scaffold for tissue and bone regeneration. So what this HA can do is encourage cells to go to that area and grow in that area and replace the HA as it degrades within your body. Biosensing is also a very common application of enzyme immobilization as a whole. And here's an example of how HA is being used in one of these biosensors. Biosensors work by essentially detecting a pH change or an acidity change in the solution surrounding a given reaction. And in this case, glucose is being bound with glucose oxidase in order to create this change of acidity that can be transferred into an electrical signal and sent and read through this biosensor. Another really interesting characteristic of HA is its CD44 receptor. CD44 proteins are on the surface of a lot of tumor and cancerous cells. So this receptor helps HA bind and localize both imaging, or all imaging, diagnosis, and drug delivery to cancerous tissues without harming the surrounding environment. So in this particular case, these researchers used these nanoparticles that contained HA to bind to lung cancer cells and then eventually prevent their proliferation. HA can also be used for wound healing. As you can see here, the same size of wound was treated with cornstarch and HA containing bandages. And so you can see that once HA is included in this film, then the wound healing process is a lot quicker and a lot more efficient. The review that I completed this summer was used to supplement an experiment that I've been working on for the past few semesters on the immobilization of this test enzyme glucose oxidase onto a mesh made by hyaluronic acid and dodecylamine. And based on the different amounts of these two molecules, we can create these different pore sizes. And in these pore sizes, we're trying to find just the right balance between high loading and stability with these enzymes and high activity. So these smaller pore sizes are going to be able to entrap more enzymes, but because there are all these other molecules surrounding the enzymes, there's going to be kind of a barrier that makes it more difficult for the substrates to access those enzymes in order to react. Whereas down here, there's the opposite. So you're going to have larger pores that the enzymes are easily kind of gone through. However, these enzymes are going to be more active because they have more access to that outside environment. This strategy can be used in a lot of different ways, but here's just one particular application of note where these hydrogels with kind of heal-promoting enzymes can help aid in the wound healing process. Now, the results of this study are not yet conclusive. However, we do have some preliminary results that show how much of the enzyme is able to load onto these hydrogels. And we do that through what's called a BCA assay. So after we immobilize enzymes onto these hydrogels, we filter the solutions, and then we have kind of an aqueous solution down at the bottom that we can test to see how much enzyme is within that solution, meaning how much enzyme wasn't caught within the gel. So by deciding how much is not caught, we can deductively reason how much was successfully immobilized. And so we take a series of serial dilutions of known concentrations of this enzyme, and we react them and incubate them. And depending on the amount of enzyme that's within those solutions, you're going to see a certain color change. And these color changes are then have to do with the amount of light these solutions can absorb. And therefore, there is a linear relationship between the amount of light absorbed by these solutions and the amount of enzyme that's within them. So we create a standard curve that is based off of these known concentrations, and we can take the absorbance of our solution and track that over to see how much glucose oxidase was within that solution. And on average, we found that about 0.055 milligrams of the 0.125 milligrams that we introduced was able to be successfully immobilized onto these gels. Now, there is great potential in the ability to control the effect and power of these enzymes. And enzymes are one of the key reasons that our bodies work as effectively and efficiently and as beautifully as they do. So here are just a few ways in which society can be impacted by these reactions that enzymes can help produce. Particularly in the energy, food and material production, environment, and health industries. Now, our ultimate goal is to create a system that can utilize the work of enzymes to their utmost potential. And to do this, we're going to have to optimize the process of the creation of our systems, the immobilization technology that we choose, and the enzyme itself. And once all of these areas are combined and optimized together, I truly believe that the possibilities are endless. Now, I finally wanted to acknowledge the National Science Foundation, as well as the West Virginia Research Challenge Fund and the Statler College of Engineering and Mineral Resources for their continued help and encouragement for not only my own lab and myself, but my entire school and research as a whole. And lastly, here's a list of references that I used to create this presentation for you all today. Please let me know if you're interested in any additional materials or have any questions. I would be absolutely happy to help. Thank you all so much for listening today.